We come to the next presentation that will be given by Dr. Helmbrecht, and he's going to talk about uptake on nanoparticles, tracking, quantification of subpopulations with regard to size, concentration, zeta potential, and fluorescence. So the floor is yours.
Yeah, thanks a lot for introduction. I'm going to talk about in the next 12 minutes about another technique how to analyze nanoparticles of various kinds, not only nanoparticles. You also can, of course, analyze gold or silicon dioxide, liposomes, any kind of nanoparticles with the nanoparticle tracking analysis. And one of the benefits of this technique is it's a multi-parameter instrument, and we're going to show what it can do. I'm representing the company Particle Metrix. On the very left side, this is the particle tracking instrument, but we also have DLS and stability titrations, mainly based on the electrophoretic mobility, but this talk is on particle tracking. So what is particle tracking? Basically, it's quite straightforward. You inject, you visualize particles. It's a microscopic technique, so you directly see your particle and what is going on. Basically, you look at the Brownian motion and you do an analysis of the Brownian motion, and you get out data from the migration behavior, the diffusion behavior of the particles, as well as size and concentration and more. So this is how it works. There is a laser, and this laser illuminates the particles, and basically the reflective light of the particles is analyzed, and this is directed on and magnified via an objective on a video camera. And by doing that, you can get out the size, the count, which is the concentration, and zeta potential, basically surface charge, and fluorescence if you want to apply antibodies which they bind specifically onto your nanoparticles. So just browsing quickly through the parameters, I guess most of you are familiar with the single parameter size. The size range distribution is relevant, not only the average parameter, but the full distribution. It gives you basically an idea if you have a homogeneous sample or if you have more subpopulations regarding a size, such as aggregation. Concentration, you can measure the particles per ml, which gives you an idea about the doses. And for example, you can do the viral titer, or you can measure the number of liposomes you have in a specific volume. The zeta potential surface charge, it gives you an idea about the charge of your nanoparticle, and differences in zeta potential of uncoated and surface modified, let's say, gold nanoparticles. They can act as detectors for some biomolecules, and they change zeta potential. Fluorescence gives you biochemical discrimination depending on the dye and the antibody you're using, and so you have a multi-dimensional measurement with this NTA. And it is not only good for bio-nanoparticles, we also do emulsions, proteins, virus, and metal nanoparticles until to nanobubbles. So how is size measured? We look at the migration, the Brownian diffusion of the nanoparticles. In this case, this is gold nanoparticles, 40 nanometer, and we record an image series. And with an algorithm, with a digital algorithm, we can detect and localize the particles. And the movement from frame to frame is depicted in the terms of the average mean square displacement, basically the jump of the particle from one frame to the other. And happily, Einstein did the calculation over 100 years ago how to do this in a 2D plane. And with the Stokes-Einstein equation, you can calculate the radius of a single particle. So this is a single particle technique. And we also do mixtures, so there is resolution, and you also see the concentration and the of such samples. This was a mix of 100 and 300 nanometer, and you get out resolution and concentration. It is crucial, as it was pointed out during this session, if you're measuring, it is good to have good statistic validity. What do I mean by that? This is a single particle measurement technique. In order to give distributions about size or zeta potential, it is crucial that you have at least a sound number of single events. And so it's crucial not only to measure at one position and measure over and over the same particles again, but shift to different position and measure and capture a lot of particles. So with this multi-position scan, you get at least 11 positions covered, and together with the pumping of the pumps, so you renew your sample, you get a lot of independent measurements creating data sufficient for statistical analysis. So this, even if you have a complex sample, you get a representative result and statistical validity of more than 5,000 particles within minutes. I'll talk a little bit about statistics. So as we are measuring through several positions, you have the outlier controls, and they directly say if something was wrong with one of the positions, and you can keep them, or most of the time it's better to remove them because they are real outliers which can explain by statistics. So this makes the whole measurement more safe, and you're not prone to believe in artifacts. An example in real world, so this are cells. They can shed apoptic bodies or microvesicles, or the exosomes. They are the so-called microvesicles, and this is the range the nanoparticle tracking can work. And these are examples from EVs. They are isolated from human serum, and depending on the isolation, you get different particle size distributions and behavior in the peak size, for example. So the NTA instrument is capable of see such differences. Concentration, how do we do that? Basically, you see the reflective pattern of the different particles, and because we know the volume, which is around 5 nanoliters, we can have a linear relationship between the detectable incidence of the particles by knowing of the volume, and so calibration is easy. It's a quite fast technique. Practically, you can do 120 measurements a day, but you all know a measurement is no measurement, so you have to do triplicates in order to really assure what you're doing is within a certain error bar. So it comes down to 50 to 40 full characterizations per day. Reproducibility of the instrument is better than 5%. And the question you might ask, yes, you see the particles, but what about the difference in biochemical behavior? So there are different strategies you can do. You can either rely on the isolation kits or ultracentrifugation, but you also can do labeling with fluorophores, with quantum dots, which gives you much more bright light, or labeling with gold, so you detect the gold which is bound to the antibody. And every one of that has pros and cons, and I'm going with you through some examples, what you can do. So this is an example from extracellular vesicles from a HEK293 cell line, and there was an antibody to a certain protein specific, and the fluorophore was Alexa 488. So this works quite nice. So, and just please apologize for that. So this is a more physical, rigid point of view. So you have your particle or your vesicle, and then you have the antibody, and the fluorophore is on the antibody. And by illuminating that and exciting that, you have the fluorescence. And by the use of a specific filter, you filter out the scattering light, only transmitting the fluorescent light. And so you can do the fluorescent NTA, and the power is in the antibody, and this works quite nice. There is also high resolution in fluorescent mode. We can also, in order to increase this, we can make use of multidimensional and multivariate statistics, not only taking into account the step, the jump of the particles, so the diffusion behavior, but also the size, so how large is the object, and parameters, physical parameters of the object, so we can do multivariate statistics in order to separate. So this was quite straightforward. Because it's two different sizes, so this is not so difficult to do, but just to give you an idea what you can do. So there is resolution because we make use of a lot of dimensions you have in an image. So there is resolution of 100 and 200, and you have a lot of information which you can make use of. This is an example for ACM-derived extracellular vesicles tagged with the dye PKH67. And by doing that, you really get an idea that not all of the particles you really see are vesicles. So this is, in our trials with the customers, we saw roughly 10%. So something around 10% of the particles are actually EVs. So what to do if you have mixtures, complex mixtures? So this was done with a 16 nanometer gold and polystyrene. If you analyze this video right away, you get one single size distribution. But if you make use of this information, which is in the image, in this case, this is the intensity and the size of the objects, you really can separate the polystyrene from the gold because the refractive index is of such big difference. And this gives very well separated populations, and you can really make a separation of these two distributions. And of course, this is the proof of principle, polystyrene and gold, but you can also use, of course, different refractive index particles of the same size, and you can separate them. A word about Zeta potential is in the talk before we heard about this concept. So this is basically the migration of the particles in an electrical field. In this case, you can really visualize this, and this has been invented, so to say, or discovered over 100 years ago, and the geochemists, they made use of it 100 years ago. And because of the tracking algorithm is quite straightforward to get the electrophoretic mobility, which is the experimental accessible parameter. And by that, by having a model, a physical model, in this case, we're using Helmholtz-Molokowski, but with our instrument, you're free to use the model of choice for your particle. And this is very nice to make characterization of liposomes. And in this case, the red curve was just the fresh liposome with no ultrasound. And as soon as you have ultrasound, so the blue one was before ultrasound. You see that you have a peak around 200, 300 nanometers. And as soon as you make a nice homogenation, you increase the concentration of the small ones because the larger ones break up. And the Zeta potential is stable over this process. So this is a multi-parameter technique you can do the size, the count, Zeta potential, and the fluorescence. And with having said this, I'm happy to receive your questions.
Thank you very much. Perfect timing. Is there any question from the audience? Yes, please.
Okay, I really enjoyed your talk, and I'm curious about using your technique to measure non-spherical particles or antisymmetric particles, which often, quite useful in nanomedicine. How do you think about it?
Yes, concentration, Zeta potential fluorescence should not be a problem. It's kind of challenging if it goes to the real size determination. Because the approximation we are doing here, we think idealistically it's a sphere. If you know a little bit more, let's say an aspect ratio, if you have a needle, we might be a little bit closer in the approximation of the size. But I would be more than happy to talk in detail, because we use this multi-parameter statistics. We can see the spherical from the non-spherical, which is a first approach. If it comes to more complex, like if you have different sizes, shapes, irregularity can have a lot of dimensions. With a little bit of a priori knowledge, we can go to more detailed answers.
Okay, final question. Barbara, please.
So if you say size, what do you mean? Is it the core size or is it the hydrodynamic size?
It's the hydrodynamic diameter, because we are in the liquid, and we see basically if the differences of electron microscope and particles, let's say from gold, this is very typical. If you measure gold in a liquid, in a medium, mostly it has a little bit larger diameter, and the concept of hydrodynamic diameter in our experience is more in the range below 100 nanometer is of more effect. As soon as you have larger particles, depending of course also on the coding, but definitely it's the hydrodynamic diameter what we are measuring.
Yeah, because in nanomedicine we can use very big polymers to stabilize the particles.
Yeah, and so then it has effect also depending on the length, of course. Yeah. Thanks for the comment.
Okay, thank you very much again.